Yeah, today I will be presenting our paper on enhancing the performance of porous silicon biosensors. So in the field of biosensors, especially in the context of medical diagnostics, our goal is to design a device that is cost-effective and also simple, that is also highly sensitive, since many disease biomarkers lie in the extremely low concentration in the nanomolar range. So in this work, our aim is to develop a biosensor for detection of a protein called lactoferrin, and we want to detect it in gastrointestinal fluids, since this protein is a potential biomarker for diagnosis of gastrointestinal inflammation, such as inflammatory bowel disease, due to its important role in the defense mechanism of the mucosal surface of the intestines. So in patients with IBD, its concentration can rise to exceeding to 10 nanomolar. And in our previous study by our group, we developed oh wait, my camera is not working. Sorry for that. So in our previous study by our group, we have developed a porous silicon-based biosensor to detect protein biomarkers in gastrointestinal fluids, and it showed great potential in handling this viscous, turbid, and highly complex sample without any pre-processing, filtering, or whatsoever. But the sensitivity of this biomarker still not sufficient for clinical applications. So in this work, as I said, our goal is to develop a biosensor for detection of lactoferrin in gastrointestinal fluids using porous silicon as the optical transducer and aptamers as the bioreceptors that is highly sensitive. So what is that porous silicon? So it is a sponge-like material with nanometric holes. It is a cool and ideal for medical diagnostics due to its low-cost fabrication and large surface area, and its ability to detect various targets label-free. So now, how does it work? So we shine light on the porous silicon surface, and we measure the reflectance spectrum. And by applying fast Fourier transfer, we get a single peak at a value called EOT, effective optical thickness, or 2nL, where n is the refractive index of the sample, and L is the constant layer thickness. So we functionalize the porous silicon surface with capture probes, and when the target molecule infiltrates and binds to the capture probes, the refractive index of the sample increases. So we see a red shift in the reflectivity spectrum and a shift in the EOT peak, which we monitor. So the primary challenge in porous silicon-based biosensors lies in the mass transfer limitation, which impacts the sensitivity. So the target analyte must diffuse in the bulk solution toward the pore entry and subsequently into the porous layer to eventually interact with the capture probes immobilized on the surface. So we can overcome the hindered diffusion within the pores by tailoring the nanostructure of the porous silicon. So we have already built in our lab a comprehensive model that explains the mass transfer phenomena in porous silicon. And I will not go through it, but it shows that the binding rate increases for larger pores. And based on this, we produced a pores with diameter of either 50 nanometer or 80 nanometer. Keep in mind that lactoferrin is a large protein, so we produced large pores. It also shows that the binding rate increases within a thinner porous layer thickness, so we fabricated layers with thicknesses varying between 5 to 3 micrometers. So this figure shows the net change in the relative EOT after 1 hour of incubation with lactoferrin, which attributes to the accumulated amount of lactoferrin within the pores. So we saw that the pore diameter has a significant impact. So in the case of 50 nanometer, we saw that there was no significant difference between both layer thicknesses, which indicates poor lactoferrin infiltration in comparison to the 80 nanometer. We show also that for the thinner porous layer of the 3 micrometer, we got the highest EOT signal, which is attributed to the shorter diffusion length and the higher concentration gradient, which is the driving force for diffusion. So, but also when designing a biosensor, an optimal a capture probe density is crucial to avoid any steric hindrance effect and enhance the overall performance of the biosensor. So we chemically modify the oxidized porous silicon to immobilize a lactoferrin aptamers targeting lactoferrin using a standard carbonamide chemistry, and we tested different concentration of aptamers. And as expected, increasing the aptamer concentration resulted in enhanced response, and we got the highest response for the concentration of 10 micromolar, and further increasing the aptamer concentration resulted in impaired aptasensor performance. So we tested the aptasensor performance by introducing different lactoferrin concentration in buffer solution, and it showed amazing sensitivity with a limit of detection of 50 nanomolar, which is already 10 times lower than previously reported porous silicon-based aptasensor with a thicker porous layer. But as always, life is not easy. So when we moved to real biological sample, gastrointestinal fluid, the limit of detection was calculated to be 600 nanomolar, and it's attributed to non-specific interaction of non-target biomolecules that interacts with the porous silicon surface. And this is another topic. We had this challenge in our project that I will not be discussing today. Now I will be moving to present the integration of the resulted biosensor in sophisticated 3D printed microfluidics to overcome the bulk diffusion. So as I mentioned earlier, an additional limiting factor is the bulk diffusion due to the rapid formation of a depletion layer that at the pore entrance that impact the biosensor performance. So by applying convection, it will result in a constant concentration at the pore entrance, and it will eliminate the formation of these depletion regions. So this study is in collaboration with a group of Professor Janina Bannemann from Leibniz University of Hanover, and we proposed two different 3D printed microfluidic designs, and it incorporates micromixer elements. So the first design consists of a staggered herringbone mixer, and it consists of two repeating ridge pattern that alternates along the microchannel, and it will induce mixing by creating chaotic advection. So here you can see this is the bottom view of the device where the porous silicon is integrated, and here you can see the cross section. So here goes the porous silicon, the microchannel, and the inlet and the outlet. So the second device consists of a three-blade microimpeller that is accommodated within a mixing chamber above the biosensor surface, and this design is studied here for the first time. So the design process was supported by flow velocity profile simulations, and it incorporates particle tracing with the same size as lactoferrin, and the interaction chance between the target and the surface was calculated. So as you can see for the SHM structure, the ridges disrupted the parallel flow, and also it accelerated the particle movements underneath them, and the calculated target surface interaction increased to 14% in comparison to no interaction in comparison to a plain microchannel with similar dimensions. But also adding the active mixing using the microimpeller system, it accelerated the particle movements, and the interaction chance even increased to 65%, which suggests that the greater potential in enhancing the biosensor performance. So for the biosensing results, the integration of the SHM was found to induce some enhancement in the optical signal and reduce the limit of detection to 20 nanomolar, and it is better than the plain microchannel as expected. And by using the microimpeller system, we saw a notable enhancement in the optical signal, and we even could have sensed a lower concentration that we couldn't sense with the other system. And we succeeded to lower the limit of detection to 3 nanomolar, which is a clinically relevant concentration. And these results also correspond well with the simulation results. So as a conclusion, we show that enhancing the porous silicon biosensor performance relies on both convection and to enhance the mass transfer to the pore entrance, and also on the nanostructure design to enhance the diffusion within the pores. We also showed that we succeeded in lowering the limit of detection from the micromolar range to the nanomolar range. So thank you so much for listening.
So good morning, good day, good evening. It's a great pleasure for me to contribute here to this fantastic series of talks in the field of microfluidics. Just click my own camera here away. Okay, and I'm happy to take this opportunity to introduce you to topics where we are currently working on, all in the field of assessment of drugs. And I will show you two platforms. One is droplet networks. It's basically droplet interface bilayers, and the other one are droplet arrays. And I want to start very simple and show you what actually is important in the drug development pipeline and testing pipeline. So if we want to have a drug, of course, this involves many disciplines and many aspects. And at the beginning, there is a synthesis of the drugs, so they're creating a library, making a lot of different drugs, and then analyzing the different aspects of the drugs. So a simple analysis is if the drug is actually stable in water, if it's stable at different pH, if it's soluble in water, and so on. And then once we have this basic, like say, chemical assessment, of course, it's important to know the drug, how the drug is actually affecting cells and tissue. And first of all, of course, it should have an effect on specific cells. For example, on cancer cells, let's say, it should kill the cancer cell, but not have a harmful effect on the other surrounding tissue. And for an infection, you want to kill certain bacteria, but maybe not all bacteria that we have in the body, in the body. So we will analyze, we can analyze, or we should analyze this very early on to assess a drug. At the same time, also we can answer, or we should address the question how surrounding tissue, how other organs in the body would be affected by the drug, if there are side effects and so on. And yeah, we will have at the very beginning, of course, models for this in order to find out if there are these adverse effects. And then finally, of course, this is a medical field to really test the drugs first in animals and later in human beings and see the effects, the wanted effects and the side effect of the drugs. So all in all these procedures, I think there are many opportunities for microfluidics to contribute here to improve this drug assessment and to make it faster or more efficient, but also to look into certain details of the drug properties and provide models that help to assess drugs. Not only new drugs and finding or searching for new drugs, but also testing existing drugs and see how they actually which effects they have on cells and the body. So in my group, we develop such model systems. We have different systems, and most of them address certain aspects in testing a drug. So from very simple model systems, where we can actually set really precise conditions, so we know exactly what our system is made of in terms of the drug, in terms of the environment. And then we can, in these simple systems, see certain aspects and also simulate these aspects. For example, permeation, this is what I will show you. But then we also develop other models, and the more complex the models are, just like having certain aspects of dynamics of the drugs, or also then seeing interactions of drugs with tissue, with cells, and so on. The more we make these models, the more complex it is, but of course, the more we also model real conditions. But then we don't have a full control of these systems, and we have more variations in the outcome. Yeah. So I'm going to show you today two platforms, and the first is showing droplet interface bilayers and how we use them, and the second is addressing high throughput drug testing, and for this we used microdroplet arrays. So one of the parameters that are assessed for every drug very early on is the capability of the drug to permeate membranes. Most drugs are administered orally, and they have to pass several membranes in the body until they reach the site where they should be effective. So the question is, how does this drug can enter actually cells or cross cell borders? And there are different ways to do so. So a drug can passively diffuse across a membrane, but there are also membrane proteins that help some drugs to translocate from outside to inside of the cell, as well as there could be carriers like lipid nanoparticles that help to bring the drug into the cell. We are mostly interested in passive diffusion, so really just the process where the drug just translocate across a membrane without any help of membrane proteins, just crossing the lipid border. And for this, to analyze this, we need the lipid membrane, and this we use or we assemble by synthetic lipids. And we separate two compartments, one compartment that carries the drug of interest and the other one, so this is what we call donor, and the other one is what we call acceptor. And then we look after certain incubation time, how many drugs actually cross this border to analyze how fast they do it and if they are permeable or not. So for this system, we use droplets. We create two droplets. Both of them has a monolayer of lipids at the interface to oil. And at the point where the two droplets come in contact, they form a droplet interface bilayer, or also called dip. So that's used quite as a standard nowadays. So this is almost established system in this field. In our case, we also separate these droplets at a certain time, after 16 hours usually, and then analyze these donor and acceptor droplets separately. I show you on the next slide how this micro device looks like. So it's one device that has many cavities. In these cavities, we can pipette the droplets, and all is surrounded by oil. And then it has a bar or a central piece in the middle that we can move up and down. And also on this side, we have cavities for the droplets. So when we move this bar, we look through a microscope and can see when we actually pair two droplets. And these droplets immediately form a droplet interface bilayer. This is shown in that video here. You see immediately the straight line here, and this indicates the formation of a bilayer. It's fairly stable, like for the 16 hours that we use for incubation, but it's not so stable that we cannot disconnect this, and this you can see in the other video where we separate the droplets after this incubation time. And then we analyze both droplets individually by use of an LC system that is coupled with absorption measurements or with MS. So this important part here is that it's label-free, while typical permeation studies are done with a label to the drug, and then we use fluorescence microscopy. Our system can assess the drug permeability without any labels. I show you now how these results look like. So this is a chromatogram that we get. And we do make a chromatogram from the original drug mixture as well as the mixture that we have in the donor droplet after the incubation time, as well as in the acceptor droplet. So if you look to the first peak here in this chromatogram, we see that there is a green line and the dashed line. So this indicates that the mixture didn't change in the donor droplet. In other words, this droplet, the drug in this droplet did not permeate. So it's impermeable, and we give this a permeation classifier in 0. But there are other peaks like this one here, where you see there is a change, and you have still more drug in the donor droplet and less in the acceptor droplet, but it indicates that the drug is permeable. And this we say it's intermediately or partially permeable, and we classify this with just a number, but this helps us to do the classification, and this is 0.5 here. And now you will know we have also a third class here. So we have acceptor and donor droplets with the same peak height. This means after 16 hours, the drug equilibrated on both droplets, and we classify this with permeation classifier 1. So having this system here set up, we can analyze a lot of drugs As you can see here, we put many drugs together and analyze them just in one experiment. And we did this for a drug library where we had to remove a few drugs because some are not stable and some actually dissipate directly into the oil. But for many drugs, it were more than 70 in the end, that we could analyze and classify into impermeable, slightly permeable, or highly permeable drugs. And this aligns fairly well with the hydrophobicity, or in our case here, indicated by the retention time of the drug in the LC system. It's not only a measurement, an endpoint measurement. In principle, we can set this up in the way that we can also do dynamic measurements, as shown here. So we can separate the droplets earlier than the 16 hours and get time points at earlier time points. And by this, we can get a kinetic curve here. As you can see here, in the donor, the drug goes down. In the acceptor, the drug concentration goes up until they meet and equilibrate. And from this kinetic curves, we can derive the permeability constant, as you can see here with the equations that are given here. Yeah, for this example here is doxycycline, and we have a concrete value here. So we went on from the drug library and selected drugs that are specific for a disease. Yeah, so while the library were just a summary of drugs that are used for different diseases, we selected here only drugs that are used for infection, so antibiotic drugs. And we choose here the set of drugs that we add in our first droplet as a mixture. And you see it was, it represents very well like a library with impermeable, slightly permeable, and highly permeable drugs. And here we wanted to analyze what kind of parameters actually will change the permeability of the drug. We looked first to the temperature, yeah, as you can see here. So we heat up our system to 37 degrees Celsius. And what we observe is that there is no change in the permeability for these drugs, which can be expected because an impermeable drug will not be suddenly permeable just by heating up the system. We then also changed the membrane composition. Here it's addressing antibiotics, so it made sense to use an E. coli representing membrane. So we used an extract, lipid extract from E. coli, and analyzed the drugs again and could see that there is again no change in the permeability. The intermediate drugs here were unfortunately not stable enough because during the formation of this E. coli membrane or dip, we had to heat up the system much higher than the 37 degrees, and the drugs degraded, so we couldn't analyze them here in this particular experiment. But what we could do is we analyzed also all the drugs by setting up a pH gradient. So one droplet contains pH 6.5, and the other, the acceptor droplet, has a higher pH. And then what we can see, this pH gradient influence actually the permeability. So one of the drugs, this one here, this is rifampin or rifampicin, this has a higher permeability when there is a gradient, a pH gradient across a membrane. And what's also nice with the system is not that we only have these clear parameters that we change, but we can also now add or functionalize the membranes that we have in the between the two droplets. And we show this by use of alpha hemolysin. This is a membrane pore that is fairly easy to use. So we just add this to the droplets, and it integrates into the membrane and forms the pores by self-assembly. And this pore allows now also molecules to cross the membrane, even if they are not permeable by passive diffusion, but they can just translocate through the pore. And that's indeed what we can see here also. Both drugs that were impermeable beforehand became permeable, and also the slightly permeable drug became highly permeable here. Just rifampin did not change, and the reason is that this drug is very large. Too large for the pore, it cannot pass through it, so there was no change in this experimental setup. Yeah, so these droplet interface bilayers proved very useful to study really in detail the permeation of the drug and get some information of the parameters that influence this permeability of drugs. Permeation is one parameter that is of interest, but also we can connect our system to test also the efficacy of the drug inside our droplets. And we did this and continued for antibiotic drugs. And the principle is shown here in the sketch. So we have a compartment that is almost cell size. This kind of serves as a replacement for a cell, an artificial cell. And in this compartment, we add bacterial cells. So we mimic here an infection, and more precisely, we mimic an intracellular infection. And then we have in the other droplet, again, the antibiotic compound. So the idea here is that the compound passes the membrane and directly has the effect on the bacterial cells, in other words, kills the cells in the neighbor droplet. And yeah, it shows the first experiments that we did with doxycycline in one of the droplets. The other droplet is filled with the bacterial cells. We wait a couple of hours just to come to the point where we have the exponential growth phase, and then we bring the droplets together. The drug will diffuse across the membrane, and we see or we can observe the growth of the bacterial cells. In our case, they produce the green fluorescent protein, so we conveniently can analyze this by fluorescence microscopy. The curves are shown here in the graph. So you see a different height at the end. This depends on the concentration of doxycycline that we fill in the donor droplet. The more we have, the less cells are surviving. And at some points here, the intermediate curves here, we can see that the cells are not growing much anymore. Yeah, they have a lower suspension and lower concentration in the droplet at the end. So this shows that also the effect of doxycycline, that it's bacteriostatic and prevent cell growth while not completely killing the cells. With this system, we can also analyze what happens when we withdraw the antibiotic compound again from the cells. That's exactly what happens also in the body. So we clear the drug from the cells at some point, and also it's secreted from the organism at some point, or from the patient at some point. Yeah, so this reduction of concentration of the drug we can also simulate by replacing the droplet that we have initially, where we have the drug inside, with a droplet that has only buffer. Yeah, you see this here in the line here. So we start with a bacterial culture that we expose to a drug, and it equilibrates for a while, and then we replace this with water or with buffer droplets, and then it gets another equilibration, but in this time the flow of the drug goes outside the droplet where we have the bacterial cells. And we do this repeatedly so that we have a low concentration of the compound in the end in the droplet where we have the cultivation of bacterial cells. Yeah, so because we know the kinetics of doxycycline, I've shown you this a couple of slides beforehand, we know exactly the concentration of the drug that we have in the droplets at any time point. Yeah, and this gives a dashed line here in the graph. And we observe at the same time the growth of the bacterial cells, which is stopped at the beginning due to the high concentration of the drug. And then at some point when it's cleared and the concentration is below a certain concentration, much below the MIC value, so the minimum inhibitory concentration of the drug, then we see a regrowth of the cells in the droplet. We also can study profiles of drug doses that a patient would get. Yeah, for example, you get the drug doxycycline, you take it, it's in your body for a while, then it's cleared, the concentration decreases, and then you take a second dose of the drug. And this we do also by a sequence of droplets that we add to the first droplet. So first we add the droplet with the drug, wait for an hour, clear it with a droplet that has just the buffer inside, and then add another droplet with the drug at a higher concentration again. So what we see here is when we have the first droplet with the drug, we inhibit the growth. So this is again the bacteriostatic effect of doxycycline. Then we have regrowth when we dilute the drug concentration in this droplet. And when we add another droplet with the drug, we see, surprisingly, again, a growth of the bacteria cells at this point. And we believe this is due to an inoculum effect. So at this point, the number of cells is very high already in the droplet, and higher concentration of bacteria lead to a different effect of the drug. So you need higher concentration to have the same effect. Finally, we can also form networks of these droplets. So we couple many of the droplets together, and between all these droplets, we have, again, a droplet interface bilayer. Yeah, we see this here for 5 droplets that we are connected, and in between you see you've seen the droplet interface bilayer, and at some point we can also, again, remove the droplets and disconnect the droplets if we want. So this simulates basically a tissue where the drug has to go and diffuse through several membranes until it reaches like deeper areas in the tissue. Yeah. So in our experiment, we used the first droplet to fill it with doxycycline, and the other droplets are all filled with bacterial cells. I'll show you now first a video here. What you see left side is a drug-filled droplet, and the other droplets have the bacterial cells. And you can immediately see that the first droplet that is most close to the drug-filled droplet has a lower fluorescence intensity, indicating that the cells are not growing well there, while the others, particularly the last one, is not much affected by the drug. Yeah? Although it diffuses over time, there is a higher concentration of bacterial cells in these droplets. Again, now we can use fluorescence microscopy to analyze the growth of the bacterial cells, and you see the lines here showing exactly this, what I just described, that the droplets at the very end are not affected by the drug. The beauty in the system is now that we can very precisely model everything that we see here. So we take into account the lag time of the bacterial cells. We know the normal growth of the bacterial cells, and we can add in the equation a part that describes the inhibition of the cell growth. And taking this all together, we can simulate exactly what we see in our system. And as you can see by the lines, particularly not the dashed lines here, these black lines here, they fit very well to the observations that we make. So the system and our simulations is really a very good and precise fit. Yeah, some residuals deviations may come from the fact that we cannot fully quantify the number of cells that we have at the beginning, plus the area of the dip is, let's say, also an approximation and not a very precise area that we define here. So at this point, I want to finish here and just summarize that we use dips, and these are very viable models for cell membranes. Our experiments with the drugs also indicated that we have really the same results, or very similar results to alternative methods that use cell membranes and analyze the bioavailability of the drug. It enables to precisely model passive drug permeation and allows us to increase the complexity of the lipid mixtures in order to find out how this influences the permeability of the drug. And we can also add in future, this is a goal, to add membrane proteins to analyze in more detail what membrane proteins would actually affect this permeability. Okay, with this, I want to continue with another platform and slightly move on to a slightly different problem. Now, testing drugs directly on cells, seeing the effect of the drug on the cell. And in many cases, as we have seen before, for infections, and we stay with this, the effect of the drug is really killing just killing the cells. Yeah? So we stick to this type of drugs where we want to remove bacterial cells and kill bacterial cells. In order to test drugs on bacterial cells and or other cells, you would usually need a platform that allows you to do many cultivations. And for this, usually people use multi-well plates. So there you have many cultivation chambers, and you can add the drug by manual pipetting, or if it's a lot that you want to do, you can also use pipetting robots. And if you have really a lot of drugs you want to test, or really a lot of conditions you want to test, you use automated systems also to handle many of these multi-well plates. Yeah, and then the growth can be just monitored by microscopy. So this consumes a lot of plates, a lot of equipment, a lot of expensive equipment also, and it consumes also a lot of cells and compounds. You need a lot of compounds to do the test. So usually multi-well plates are filled in with volumes of microliter per wells. So here, microfluidics can also help to miniaturize these wells and to produce compartments that are in the range of nanoliters or even picoliters. So this gives a massive decrease in the volume per compartment, which also means you need much less drugs and, of course, less cells, less medium, and so on. It also helps to make it faster because these compartments can be very close to each other. And in this way, you can analyze very quickly by microscopy, just taking pictures of many compartments in one picture. And there are different ways and microfluidic methods to make these small compartments. That's very common. Either you can use droplet microfluidics, a very common method here, right, to make these droplets in a channel system, and then you have nanoliter or picoliter compartments where you can enclose cells. Or you can also engineer systems with small compartments, like use of valves or use of walls, basically a decrease of the multi-well plates, making really just smaller wells here. Now I want to introduce you a platform that we use as a replacement for multi-well plate assays. And this is based on droplets, but it's an open system, and therefore we call it open droplet arrays. So in this system, we have droplets on an open plate. The plate has a size of a microscopy slide, so it's a bit smaller than a multi-well plate. And the plate has a pattern surface. So on the surface, we have hydrophilic spots. So these hydrophilic spots host the aqueous droplets with the sample. And it's surrounded by a hydrophobic surface, which helps also to cover the entire plate by an inert oil. And the droplets stick here, so they don't move onto the areas where we have the hydrophobic surface. It's a glass plate, so we can do all microscopy that we are used to and that we need to analyze cell growth. But it's also open on the other side, so it gives us opportunities to couple it to other microfluidic, to other analytical detection systems. And we use mass spectrometry, particular MALDI MS, to analyze the sample as well. In MALDI, you have a laser that ablates or shoots on the sample. There is a matrix that absorbs this very heavily, and this helps to ablate the sample. And you get analytes and you get ions that you can detect then in the mass spec instrument. So this system is very close to what you have also on a multi-well plate, as you have index droplets, but it's just much more dense. It's similar to droplet microfluidics, and the number of droplets we can host on a cell is very high. So we made plates up to 100,000 of these droplets, still have on the size of a microscopy slide. So very dense systems. One word to the plates that we use, because they are also essential here to build up the droplet arrays. We use a glass slide that we cover with a conductive material called indium tin oxide, ITO. It's a very thin layer, so the plate is still transparent, and we can see the cells when we use a inverted microscope. We coat this with polysiloxane mostly, so this gives a hydrophobic surface. And by photolithography, we remove parts of this surface coating to have hydrophilic spots here. The The plate design is, of course, as we design it, we can have hydrophilic spots with diameters that are roughly, or we use roughly around between 30 micrometers and 300 micrometer spot size. Of course, the positions are defined, and also the space between these hydrophilic spots. We can also bring them close together if we want this, and bring or make contacts between droplets. And of course, we have a defined number of these droplets on one glass slide. The plate is then placed in an oil bath, and this prevents evaporation of these tiny droplets. So they stay stable for a longer time, for hours up to days. If we even place a foil on top of this, we prevent really the evaporation of the droplets. So we have now the plate, and how we do it is shown in this picture here. So we use the sample, we have the sample in an Eppendorf tube that we pressurize. So this is a pressure-driven pump, basically. And by this, we transport the sample through the capillary onto the plate. The plate is a motorized stage, so we move the plate underneath, and by moving this, the droplets are formed. I show you this on the next slide. Just want to show you the picture or the sketch again, that this is just all on a standard microscope. Yeah. And here's now the process, how this works. So fluid flows from the top through the capillary. There is a small distance between the end of the capillary and the plate. And then when the fluid flows and we move the plate underneath, we can see that the water, or this aqueous solution, just shears off and releases a droplet on the spots. And as you can already see in this video, these droplets are really very homogeneous in size. So the flow that we have in the capillary, together with the movement of the stage, the speed of the stage, defines also the volume of the droplet here. So it's easy to do it first time, and it's not more difficult to do it a second time, because we can use the existing droplet as a hydrophilic anchor for the fluid and do the same again. So another droplet can be positioned on the already existing droplet to add compounds or medium or whatsoever. Yeah. And this is very fast. So I give you here some numbers. A plate with 7,200 droplets can be created in 12 minutes. And then most time is needed to actually change the direction of the moving plate. So it's not linear. When we have a larger plate, so this is more than 3 times larger, we don't need 3 times more. It's just 22 minutes for this much larger plate. And it's not only possible to look then through a microscope to analyze the droplets, but as I mentioned already, we use mass spectrometry. We read out these large arrays by placing the plate into a mass spec. So for this, we have to remove the oil beforehand, but this is very fast. Plus, we have to deposit a matrix compound on top of this. We developed for this a custom-made sublimation chamber that works very well to distribute these compounds evenly across the entire plate. And then our system that's made for tissue imaging, it's a very fast system and allows us to read out the plate of more than 7,000 spots within 1 hour. Yeah, so this examples here that you see is just one molecule, yeah, angiotensin 2 labeled with actually fluorophore that we just need to for visualization or useful visualization. But this plate here shows you really the multi peaks and intensity of the multi peaks, and you see it's fairly evenly distributed all across the plate. Yeah, so each pixel here represents one droplet on the plate. The process is very well controlled, and I already said that the flow rate determines also the to good portions the volume of the droplets that we have on the plate. This is visualized here. So if we on the plate just decrease the flow of the fluid, we get less and less volume inside the droplet. Yeah, and so on one plate we can create droplets of different volumes. And we proved this by use of confocal microscopy to visualize the side view of the droplets, and you can see this in the images here. The higher the flow rate, or given here as a pressure on the Eppendorf tube, the higher it is, the larger is the droplet volume that we get onto the plate. And this change in droplet volume, we can also exploit to form gradients. So if you spot one compound in this way, and then you add another compound or just buffer in the second run to fill up the droplets and get the same volume in the end, you can create in this way gradients all across the plate, as you can see here in this image where we just used fluorescein. Of course, you can make smaller gradients as well if you want. So the same plate here, and here you see several lines give one gradient from 0 to high concentration of fluorescein. And before I come back to the drug testing, I show you one example where this can be exploited. This was our test example to also establish a platform again. Here we analyze an enzymatic reaction in this without the need of fluorophore. We used a peptide and we used also fluorescent tech, but this was also again to visualize that we have the gradients on our plate. So this peptide that we use can be cleaved by an enzyme, and the product is a smaller peptide that we can analyze and differentiate very well by use of mass spec. So what we do is we spot the different concentrations of this peptide on the plate several times, yeah, and then we add the enzyme, but the addition of enzyme is done at different time stamps, different time points, yeah. At the end, after the last spotting procedure, we remove the oil, dry the droplets quickly, and in this way stop the reaction for the entire plate. So we have different concentrations and plus different time points, which is enough, sufficient to do a kinetic analysis of the reaction. And we did this not with fluorescence microscopy, as said, we did this purely with mass spec analysis. And this is shown in the spectra that you can see here. So for short reactions, there is not much time to build the product. So we see mostly the substrate and very little of the shorter product. But then over time, we see mostly the product, the shorter peptide, and nothing of the original substrate. With these different data here, so different colors represent here different concentrations of the enzyme. With all this information, you can calculate the kinetics of the reaction, which we did. It's actually just simple Michaelis-Menten kinetics. We could derive parameters for this particular reaction. Coming back to cell analysis and drug testing, yeah, we have now a system that allows us to make large droplet arrays, that allows us to observe cells in the droplets and to later on, if we want, a detailed analysis with mass spec. And we established here a workflow where we spot the droplets, we incubate the droplets so that we can observe cell growth over time by microscopy, and then decant the oil, so remove the oil, add the matrix, and do the MS analysis. I show you first not drugs, but another example, a bit older example, where we also established the system to analyze products of cells. So in this case, there were yeast cells that produced an enzyme. As the enzyme is secreted into the droplet, accumulates there, and then at a certain time point, we add the substrate and observe the reaction. And for this reaction, we had two substrates, one that built up a fluorescent product, and one that is the actual reaction, the native reaction, and of course this is not fluorescent and does not produce any fluorescence during the reaction. Yeah, so usually the enzyme is characterized by this reaction here that uncages fluorescein. And we can do this also on the plate, of course, and see the different steepness of the lines. So this means different concentration of the enzyme that we have in the droplet. But we also can use mass spec and use a native reaction for this enzyme, by the way, that's phytase. And it cleaves phosphate groups from phytic acid until we get inositol. And with mass spec, we were able to observe all the intermediate states in this reaction, plus we could also calculate the activity of the enzyme in the droplets. So now we come really back to the drug testing. In this case of drug testing, we use bacterial cells in the droplets, and then we add the drug of interest at the same concentration to have many replicates, or the drug at different concentrations. And first of all, we just, of course, made sure that the bacterial cells grow there in these droplets. And this is the first data I show you here. So you can see bacterial cells at the beginning, so we can't really see them well in these droplets. But over the time, they grow and grow and grow, and we see bacterial cells producing GFP again, and we see how they grow inside the droplets. We used a full plate just to spot these bacterial cells, and in this way got finally more than 18,000 growth curves, showing basically all the same bacterial cells that grow in the medium. So there are some differences. These are because the content of the number of cells we have per droplet is of course not the same in every droplet. It is fairly homogeneous, distributed across the entire plate, but we have some droplets with just a few cells at the beginning and other droplets with a bit more. So between 5 and 10 is a normal starting number of cells. Of course, you can make it you can delete even more and have just a single cell in a droplet if you want. And then we used the same approach, and you added a drug at different concentrations on the plate. So we had always 120 droplets per condition, and added then amoxicillin at different concentrations. We used the concentrations that are around the MIC value. This is the minimum inhibitory concentration, and the value is given in a document from Oikast. Yeah, so at mix, all cells should die, and we mostly see this. And for amoxicillin here, we can see at an MIC of 1, it's somewhere here below, so no death. All cells are dead at this condition. And I want to say also, just look at the pictures here. We can also see the cells, and if they have some changes in morphology, which happens often when they are stressed and get antibiotic treatment, they change this morphology, and we see here the formation of long fibers in the droplet. This indicates a stress reaction of these bacterial cells. And on this plate, then we can do mass spectrometry. We have not much material. We have only a few cells in our small droplets. However, we see a good spectrum. We look mostly here into the range, into the mass range where we expect lipids. Yeah, so we look mostly to the lipid composition of the bacterial cells. Once you have this system, we can also not only use different drug concentrations, but of course you can now use one plate to study different types of drugs. So here you see here a set of different drugs. Of course, cells respond slightly differently to these different compounds. What we are interested and can do very nicely in our plate is also a combinatorial testing. So the idea on this is that an antibiotic compound can be improved or can enhance the effect by addition of another compound. Yeah, so the idea here is to find new and efficient combinations of drugs that improve the outcome of the treatment. Of course, you don't want to have adverse combinations where you have a worse effect of the drug. So this synergy testing is done on a macroscopic scale in multi-well plates by use of so-called checkerboard assays. So here you add the drug at different concentration, and the first drug you add from top to bottom, or decrease the concentration from top to bottom. The second drug you do the same, but just you decrease from left side to right side, or increase from left side to right side, so that you have some some wells with little drug, and then the outer wells with high drug concentrations. And the pattern you get, the growth pattern you get in these wells, indicate then if you have a synergistic effect of the drugs, or rather an additive or antagonistic effect. There are also ways to quantify this effect. So we started to do this, these kind of assays, and I give you one example. Again, E. coli cells that we treated with 2 antibiotic compounds, ciprofloxacin and azithromycin, and I show you how this looks on the droplet plates. So if you would spot the drug alone, you would get a picture like this. So here you have a lot of red droplets. This indicates the cells are dead, and green droplets means the cells are still alive. Yeah, at some point it turns from green to red, and this is the range where you have the MIC value. And then for the second drug alone, it would look like this. And now if you bring both on the plate together and do this checkerboard assay, you get then the combination effect of the 2 drugs. You see here that only in one corner you have still cells that are alive, yeah, while on the outside, of course, the cells are dying. And here this pattern that you see here indicates now that these 2 drugs have no synergistic effect, but more a indifferent effect. What is nice that we have a lot of data here. So we have all growth curves on the plate. This is here an 18,000 droplet plate, and we followed all droplets over the time. So we have all the growth curves, and of course we can now zoom into one droplet if we want, like one where the cells survive or one where the cells die. Yeah, so we can zoom in and selected droplets and see again the curves and look what happens there. Yeah, so a bit more, some more examples. Yeah, this is the same type of analysis with other drug combinations. This is amoxicillin clavulanic acid. It's a well-known combination of drugs, and it's well known that it's a synergistic effect. Of course, we see this also on our plate. And an indifferent effect is given by amoxicillin streptomycin, and we can also see this on the plate. And we also have preliminary data on mass spec analysis. I'll show you this very shortly here to see that we can do this. So we get spectra on all droplets of the plate, even where the cells are dead. We make spectra and then use data banks to compare the peaks that we have and get information what we can see. And by this, we identify different sorts of lipids that we find in these bacterial cells. And now our interest is to look into these lipid compositions that we find and into changes under drug treatment. And one example here shows that I start from the back side. So GFP indicates that we have the living bacteria here in this corner. And in this area, we find one lipid here, whereas we don't see the lipid much on the outer sides where we have dead cells. And there where we have the MIC value, so in the intermediate range, we see another lipid coming up. And the other, this PE26.0, we don't see here anymore. So it may give us some indications how the drug actually, the mechanism of the drug is, but it can also give us maybe new biomarkers that indicates, or we hope to find new biomarkers that indicates efficient drugs, but also developments of inefficiency or resistance. The time to end, right? Good. So my summary for this, I hope you have you've seen how nice these open droplet plates are for particular for high throughput assays. So these plates provide a high density of droplets on a small plate, on a small footprint. The droplets are still indexed as on multi-well plates, really in contrast to standard droplet microfluidics, where you often just collect them and then you lose the index of the droplets. It's fast, this process that I have shown you. It's possible to add multiple times compounds. It's also possible to select compound to remove part of the droplet of the fluid from the droplet, or withdraw a droplet just to regrow, let's say, bacterial cells on a larger scale again. Because we cover this by oil, we can do long-term studies and see really the growth curves over several hours up to days. And in contrast to droplet microfluidics, we don't need surfactant for stabilization of our droplets because they are stabilized by the surface, and we don't need at the interface between the droplet and the oil another surfactant. This is also good for mass spec analysis because surfactants actually disturb the spectra a bit. We use this platform for many, many applications. I show you some, to product analysis of cells, but then also drug testing. But there are other applications like studying enzymatic reactions. And I said before, when you can also bring droplets close together, they can come in contact, and we can also form between droplets, between droplets, some droplet interface bilayers. And last but not least, because we replace assays that can be done, or we can use them for assays that can be done on a multi-well plate. We also save the use of multi-well plates. In the high throughput area, this produces a lot of plastic waste, and it also consumes a lot of compounds. Yeah, while we go so much smaller, have much smaller droplets, and also in total we never use liters, but just microliters. We use much less compounds for our system. We can reuse the chip. Yeah, there are no wells. We can remove the droplets again, clean the chip and use it again. We can also recover the oil and then reuse the oil. So it's, in terms of sustainability, also a much better approach. Yeah, and at this point, I'm at the end of my presentation. I thank very much all group members who worked on these projects. I can almost list everyone because everyone is involved in one or the other. The first was Petra Juskova and Rob Strutt, who made the work with droplet interface bilayers. And Max did a lot on the droplet platforms together with Claudius. But other students also working on this. In particular, also alumni who started these projects really helped to improve the system over the years and make it very stable and reliable.